After I'm gonna break that. I totally knew about the Valve project. Now it's going really good. We have a full proposal together. So I introduced myself after a deluge. That's why. Yeah, you too. I like that.
I think you should.
Chopped.
It's like 5 years ago. All the hair was
I love it. I love that aspect of it. Yeah, there's.
yeah, when I have long hair.
I'm ready to see all the new triple. I think it's. I don't know why not CBS. I don't know. I don't know either. It's just one of those things my parents always had on the TV. I didn't know what channel they turned to.
Good afternoon, everyone. Welcome. It's my great pleasure to introduce Dr. Cassandra Quave from Emory University. She's an assistant professor there and has a joint appointment in the Department of Dermatology and also the Center for Study of Human Health. And she got her PhD. She's a Florida native, actually. She received her PhD from Florida International University and went on then to do two postdocs at the University of Arkansas Medical Sciences and at Emory prior to being a faculty position there. She's a very interdisciplinary research program, so I think it'll appeal to most of us in the audience here, if not all. And she's published a lot of papers, book chapters, and she's also the curator of the Emory University. And we know each other from the Staph aureus realm, so she'll be talking a little bit about that today as well. So without any further ado, here's Dr. Quave. Okay, sorry. Sorry. So thank you, Kelly, for inviting me. It's great to be back in Florida. And it was really exciting to speak with a lot of you today and learn about all the exciting research programs that you have going on here at UF. So today I'm going to talk about the work that we're doing in anti-infective drug discovery, and I'll speak specifically about a few case studies that can illustrate the ways by which we really are looking for new therapeutics for infectious disease. Can you hear my disclosures? And our overview is I'm going to start by discussing the resistance problem. We'll go over the history of plants in medicine and why on earth would I be looking to plants for new sources of medicine. We'll go over ethnobotany and drug discovery as a field. And then again, I'm going to go over some of those case studies in innovation. So talking about some of our work on attenuation of virulence. We'll talk about antibiotic initiation studies, work with classic antibacterial discovery, and then also our work with biocomb inhibitors. When I give these kind of talks to a microbiology crowd or a general medicine crowd, I like to start with a case study. And so these are Raymond and Sissy Quave. They're my parents. That's me as a baby. And I was actually born with multiple congenital birth defects in my skeletal system. And so at the age of 3, you can see I had a pretty big disparity in limb length. And so to treat this, I underwent an elective amputation. And as a result of that amputation, I also acquired a hospital-acquired infection. And it was Staphylococcus aureus. So Staph and I go way back. And I like to show this example not only to tell you a little bit about what drives me to look for new drugs, but also to illustrate some of the things that we have to really deal with when it comes to clinical care. And number one is that right now, while we currently face a rise in antibiotic resistance, this methicillin-sensitive strain that I was infected with as a child nearly killed me. It led to massive necrosis of my tissues. You can imagine a pot of vanilla jello falling out of a wound. That's what it looked like. And I also had osteomyelitis, or a biofilm-associated infection inside of the bone that had to lead to additional amputations and surgeries. So while we have problems with acquired resistance, or resistance that results from microbes acquiring genes, right, that yield resistance, we also have problems with intrinsic resistance from things like biofilms that can also lead to very poor patient outcomes. So what is the problem today? Today we face something known as the post-antibiotic era. This is an era by which we're going to have greater and greater difficulty in managing some more common infections. This is a time when things like simple surgeries and interventions may become more and more dangerous because of the lack of accuracy in some of our drugs. Some of the current estimates of the number of deaths due to untreatable infections, this is from the O'Neill report, one of the current estimates is that we currently have around 700,000 deaths per year due to untreatable infections. And if you want to put that into the context of plane loads of people, I like to put numbers in the context that I can understand. This is about how many plane loads of 300-seater jets that we lose every day currently due to untreatable infections. In 30 years, that number is projected to be 10 million, and so we're looking at roughly 4 plane loads an hour. So we have a very serious and pressing need to come up with a number of new innovations, both from the perspective of rapid point-of-care diagnostics, but also from the perspective of filling this drug discovery pipeline. And so here you can see an example of where we're at when it comes to drug discovery for antibiotics. You'll notice this big gap that starts in the 1990s. It's known as the antibiotic discovery void. In the news, you may hear about new antibiotics being approved. And the word new there is a bit of a misnomer because they aren't actually new classes. They're taking existing structures and making slight modifications to that core scaffold and rolling it back out as a new antibiotic. But as you can imagine, resistance continues to be a problem because we're not really dealing with very new approaches within these structures. One other thing that contributed to the discovery void was a transition or a shift from natural products research towards these large combinatorial chemistry libraries. The thought at the time was that if we can generate millions of compounds and test them using our new high-throughput robotic systems, that surely we will end up with lots of great antibiotics. Anyone know how many antibiotics resulted from that shift initiative? That's right, 0. Good answer. Bad thing, but right answer. So we're at a bit of an impasse now. We have a number of different reports that prioritize pathogens based on those that are at the greatest risk. Here's an example of the World Health Organization's risk list from 2017, and it ranks them as critical, high, and medium threat levels. The ones in red are ones I'm going to speak about today. The ones in purple we also work on, but I'm not going to include in today's presentation just due to time limitations. So there are also pathogens known as the escape pathogens. This came out of a paper with the Infectious Disease Society of America that are also prioritized. And you'll note that there are a number of gram-negative pathogens, and especially the carbapenem resistant pathogens are at the top of the list. So let's get back to plants. Why on earth would I turn to plants for the solution to this problem? Well, if you look back over human history, plants have served as the foundation for many of our modern medicines. These are just a few examples. things like aspirin, digoxin, cancer drugs, pain drugs, cardiac drugs, that all they were originally discovered in plants. We have on Earth around 391,000 species of plants that are estimated, and out of those, roughly 7%, or 28,000, have been documented for use in human medicine. That's fascinating to me. Humans are using 28,000 species. But if you look at the scientific literature of our real understanding of how many people have actually investigated these species in depth, I would estimate we've only looked at maybe a few hundred of those at best in a rigorous scientific way. So there's a tremendous amount of room for exploration and investigation when it comes to the plant kingdom. Now, one question I often get is, well, how did humans determine which plants to use as medicine in the first place? Well, if you live in nature, and humans are not separate from nature, we are part of nature. When you live in these environments, humans are constantly observing, interpreting, looking at animal behavior with plants. We know today that animals like, or even insects, use plants as medicine. This is an example of the monarch butterfly, which uses cardiac glycosides from a Sclephia or milkweed species to protect their offspring from parasite attack. We know that primates, non-human primates, also use plants as medicine. And there are a number of examples of the consumption of botanicals to help rid their guts of intestinal parasites. And humans are also naturally drawn to plants. This is my youngest. When I was in the field, he got into my collection of leaves. Throwing them up in the air and having a ball. But there's this curiosity, this linkage, and again, going back to observation and a lot of trial and error. Now, why are plants making these compounds? There are a number of reasons. We know that plants, while they are sessile, we know that they have very complex signaling networks at play between their different tissues. In the rhizosphere, beneath the ground, you don't just have sterile soil. You have soil that is rich in microbes and animals and fungi and bacteria and viruses and all of these and other plants. And there's all this crosstalk happening. And they're exchanging crosstalk to defend themselves, to protect their space, to attract pollinators, to attract seed dispersers. It is their way of engaging with the world. And humans have caught on to this. So here I have a slide of compounds. Does anybody know what these chemicals, I have the names here in the cheat sheet, what these compounds share in common? Two major things. Anyone recognize these?
They all made in plants. They all made in plants.
They're all made in plants. These were all originally discovered in plants or modeled after compounds found in plants. And these are all listed in the World Health Organization's list of essential medicines. This is a slide I love to show to physicians to remind them that their armamentarium, their tools that they're using, in many cases, some of these major important tools originally were discovered in plants. Now, I was trained in the science of ethnobotany. And ethnobotany is the study of how people use plants. And this doesn't just have to apply to medicine. It can also apply to the use of plants for food, for structures, for shelter, for tools in agriculture, for music, art, all these different elements. But what I really dedicated my life to studying is how people use plants as both food and medicine. And ethnobotany has also been described as the science of survival because it's through knowledge of how to use these different environmental resources that humans have learned to subsist and survive in vastly different ecosystems across the globe. I'm going to go over my drug discovery process using the tools of ethnobotany and show you how we go from the field and then hopefully someday to the clinic via the pharmacy. And we're getting really close. This is really exciting. So we start with ethnobotanical research. This can be done through primary interviews with people that still use plants as a major form of medicine. We also will look at historic texts. The world is rich in digital copies now of many ancient texts going back to the 1600s. Some of these are in different languages, but that's what collaboration with the humanities is for, where they can help you translate these. And we can look to these texts and their current day uses to help pinpoint out of those 400,000 plants, which ones should we focus on in our drug discovery efforts. We also work with botanists on our team that identify the plants, chemists that extract them and analyze them, pharmacologists and microbiologists that test them in assays. And again, we're going through this very iterative process to go from information to biological material and eventually to compounds for lead optimization. And again, what's unique about this is that we're not starting with just some random collection of plants. We're taking things that people are already using and have been using, in some cases for centuries or even millennia, to treat infection. And so we're using this as a guide to help us reverse engineer and identify chemical blueprints or molecule scaffolds that we can then do additional medicinal chemistry and SAR studies on to optimize these. So another question I often get is, how do you pick your field site and where do you go in the world? We also are very strategic in our selection of field sites. I focus on biodiversity hotspots. These are parts of the world where you have a rich number of endemic species, so species that are only found in certain parts of the world, and high numbers of plant diversity. Many of these places also coincidentally have rich cultural diversity, and the Balkans is a great example of that. So in yellow, you can see all the different sites where our plants come from. So I personally do a lot of field work in the American Southeast, both in Florida and Georgia, but also throughout the Mediterranean, especially in Albania, Kosovo, and Italy. Working with our partners in different countries, we've also expanded our collection to countries like South Africa, Uganda, Trinidad, Colombia, Pakistan, and China. And so our core research approach is really fourfold. We start with field work or field collection. We do not grow any of our plants. These are all wild collected. So if we're going based on a list, we have to go out into the woods and find these things, right? Which can be an interesting challenge, like a scavenger hunt. We deposit them in herbaria so that we have a physical copy or a record to ensure that we have the correct species that we think we're working on. And by the way, these herbaria can last for 500 to 600 years as a long-standing record of what life looked like in a certain part of the world. We then work on their chemistry in the phytochem lab, and then assess them in the micro lab. So how does this work from field to the lab? It starts with a lot of agreements, a lot of permits. We are a USDA inspected lab. We can receive materials from anywhere in the world, either fungi or plants. We have codes of ethics, international research collaborations with in-country partners. We engage in access and benefit sharing, which is something that is enforced through the Nagoya Protocol and the Convention on Biological Diversity. And this, for me, is basically starting from day one of ensuring that the communities that we're working with receive benefit from those collaborations from the beginning. We do this through a number of mechanisms, including training students and returning knowledge back to communities. So the result of this is we now have a very specialized chemical library composed of 600 species of plants used for infectious inflammatory disease. And this is, these have been used to make 1,900 extracts. Now, a single plant tissue, let's say a leaf, when it's extracted, can actually lead to hundreds or even thousands of unique molecules. So a crude extract is very complex. And then we go from that complexity and tease it apart to understand which elements are most beneficial to the desired phenotype. Importantly, though, our core mission is to have our work to be inspired by traditional medicine, but driven by bioactivity. I'm going to just show you some images from some of our field sites to give you a flavor for what some of these different environments look like, the diversity of plants. This is some work in Albania. This is in the northeastern sector of Albania. And you'll note that with this picture of the home and behind me on this photo, it's thatched roof, stones. You have animals with hand woven in baskets. Agriculture is still very much hand-driven. And so these are people that are very isolated and very much in tune interaction with their environment for not only their acquisition of food, but also their medicine. In coast development, on the other side of the border, it's very different though, because here you have paved roads, you have a lot of international development aid money coming in, and you have intensive trade of plants that were collected in these high montane plateaus that are being collected and distributed throughout European networks. Here in South Florida, this is in DeSoto County, my home county, we work with local ranchers and orange grove owners to go out into large territories that collect in a variety of habitats from oak hammocks to swampy areas and also on the Peace River, looking for plants, in this case, plants that were documented as being used in traditional medicine historically by the Seminole people. In the Agavian Islands of Italy, we work with, again, primary interviews, and I just like to include this because I am a mom. And when you have one leg and you got 3 kids you're bringing from a mountain, there's nothing better than a pack of donkeys. So this is us on our way back down off the mountain, and we have our samples kind of strapped to the animals. And grad students unfortunately have to walk. Donkeys are for the big boss. All right, and then here in just a little bit north of Gainesville, we also have a field site working in collaboration with the Jones Ecological Station at Itchaway, where we're working on, based on historic Creek Muscogee traditional knowledge and looking at plants and these unique longleaf pine habitats and collecting those. So now let's get to the staff. This is what Charlie's been waiting on. So we have this unique collection of extracts coming from plants from these different ecosystems. And one of our targets is Staphylococcus aureus, and we're going to be testing these against this bug. You know, I already mentioned how staph can be dangerous for a number of reasons, including its ability to lead to massive tissue necrosis, but also biofilm-associated infections in the tissues, including bone, wound, heart, and so on. It is a leading cause of bacteremia and sepsis. It is associated with a number of life-threatening issues. I'm going to start with a story around toxicity. So staphylococcal toxins are responsible for a number of interesting diseases. We have toxic shock syndrome that results from use of superabsorbent tampons over long periods. We have an inclusion and an overgrowth of bacteria in the vaginal tract that can then lead to expression of TSST1. Scalded skin syndrome, which includes staphylococcal exfoliative toxins in neonates. We have abscesses, necrosis, and sepsis. So these are really the hallmarks of staphylococcal infection. And then we have atopic dermatitis, which I've gotten more and more interested in based not only on field site observations, but also my interactions with clinicians at Emory. So when I'm in the field, by the way, and I'm interviewing people, this is what I'm asking them about. We're sitting down, we're talking in detail about their burn wounds, their boils, abscesses, leaky wounds, non-healing ulcers, anything that's pustulant and goosey that you're really excited about, especially when you're using plants to treat those. So in the lab, we take an approach where we really try and think about what are the right questions to ask. Historically, when you look at which questions have been asked of plants, it's usually been around growth inhibition or bactericidal activity. And those are valid questions to ask. We need to see, this is how classic novatic discovery works, we need to see, can these actually kill or slow down the growth of these bugs? But I'm also very interested in these other mechanisms, because what we have found in our work is that you can have many examples of plants that are being used consistently for the treatment of clearly bacterial-associated wounds and infections, but which have no growth inhibitory activity against the bacteria you would expect to see in that kind of environment. So then the question becomes, well, is this actually working? People say it is, their infection is resolving, but how is it working if it's not actually killing or slowing down the growth? And so this is where we're getting into questions of modification of virulence, resistance modification, and also biofilm work. You'll notice I have host-directed impact here on this slide. I know a number of you are working on host systems. I think it's really exciting. This is an area I'd love to test our plants in the future. It's just not something I'm currently set up to do. Importantly, what else we're doing is that our screens are phenotypically driven, and we do it in an unbiased way. So I'm not looking for specific classes of compounds, and I'm not looking specifically at this over new potential mechanisms and new classes that are bioactive. So let's talk about how quorum sensing actually works and how this leads to toxicity in staff. The concept of quorum quenching is this idea that if you can interfere with bacterial crosstalk, which is really important for regulation of upregulation of toxins or downregulation of other factors like biofilm or upregulation of biofilm, if you can interfere with that and basically trick them into behaving as if they were in a single cell kind of environment rather than as a group, because you're basically changing the dynamic of the population and how they behave. So one system that we've become very interested in is the accessory gene regulator system in Staphylococcus aureus. And this system works through expression of these AIPs, or auto-inducing peptides, into the extracellular environment. There are 4 known types of AIPs, and these are named, with AGR 1 through 4 based on that AIP structure. Following docking with AGRC, you have these phosphorylation events and then the upregulation of RNA3, which leads to increased secretion of virulence factors. One of these virulence factors is known as delta toxin, also known as delta hemolysin, and we have taken a method that uses HPLC, or high performance liquid chromatography, where we can directly inject bacterial supernatant onto an HPLC column and then measure and quantify precise levels of formulated and deformulated delta toxin as a real measure of virulence factors being produced. And what we've also noted is that the reduction of levels of delta toxin varies greatly from strain to strain. And we're currently working on a paper, actually it was just submitted on today, Monday. Yes, with Tim Reed's group, who has taken a GWAS approach using this delta toxin data and comparing that with some of their sequencing studies. So there's some interesting things I can't get into today, but keep an eye out for that. Okay, so when I was looking for assays to run and to try and assess some of these, one of the plants that popped up is that of Chinus herbacefolia, also known as the Brazilian pepper tree. How many of you have seen this or are familiar with it, right? Is it a beloved plant of Florida? No, it's a noxious weed. Everybody hates this plant. They hate on it so hard. They want to spray it with this phosphate and just kill it. But I say don't kill it. Don't kill it. Because this plant and its very weedy, pervasive nature may just be why it makes such a great medicine. We found records of the use of this plant going back to 1648 from a Dutch naturalist that was traveling in Brazil, and you have it continues to be in the Brazilian pharmacopoeia today, and it's used topically in the treatment of a number of different skin-associated infections. It's also used as an antifungal, but what caught my interest was this use of the fruits. They use it in balm for wounds and ulcers, okay? And in the literature, the fruits had not really been explored. And so we found these copies, by the way, in the Biodiversity Heritage Library, a great online resource for digital historic text. So what have we done with this plant? So we started with an extraction, a crude extraction. We do liquid-liquid partitioning, where you're basically separating the chemistry by different phases based on polarity. This goes through rounds of flash chromatography, and then to analytical HPLC. And you can see each level, you start to see an increase in these peaks here. And in our original publication on this, we found that we had a very clear dose-dependent response, not with growth. There's limited to no impact on growth until you get to higher concentrations, but a very clear impact on our reporter strains that were developed out of Alex Forceful's lab, showing that this is inhibiting signaling of AGR, but it is not impacting the growth. That's very important, because if you are impacting growth, you can have some false positive results. One pushback we get when you're talking about virulence inhibitors is that, well, if you know about virulence, you're going to have so much biofilm, it's going to make the infection just really chronic and it's going to really stick on. And this was something interesting that we found, is that in our biofilm models, which is a crystal violet assay, we found that, yes, at lower doses, we did see an increase in biofilm production and biofilm biomass. But at a certain dose, so in this case at 32 micrograms, we saw a complete switch. And we've worked a lot on biofilm inhibitors, and this switch from on to off doesn't typically happen. It's usually a gradual dose-dependent response. So we're not really sure what's happening here. We know something's happening where we're having this immediate switch from one behavior to another. I'm working with Alex's lab. He runs an intradermal infection model with a strain in this lab for the Los Angeles clone. This is a USA 300 strain. It's a community-acquired MRSA isolate, extremely virulent. And when you inject it into the mouse, into the skin, you can see a pretty rapid development of a necrotic lesion. Here you have at day 3 post-infection. This is day 7. And this is with a single introductory dose of 50 micrograms of the extract. You see a significant reduction in dermonecrosis. And so this was really interesting. And we thought that he also tracked this using IVIS and fluorescent reporters. This was really interesting. And we wanted to get down to, okay, we have this is the extract. It's still pretty chemically complex. Most synthetic chemists would scream if they looked at that. But for natural products, I'm like, this is clean. We got like 27 compounds. It's awesome. Because remember, my starting point is closer to 1,000. Okay? So we're down to 27 that we can really detect. And getting down to those individual molecules can be challenging. We are trying to deploy some new methods that we're developing in the lab that leverage some interesting bioinformatics platforms that are being developed out of Peter Dornstein's lab, the GNPS, and also out of Roger Millington's lab, compound activity mapping networks. Really exciting initiatives that are ongoing there. But what we did is leveraging their bioinformatics platform, we tested different related species of these and are looking for ways to highlight the features in the mass spec, the features or the compounds that are most clearly associated with the bioactivity. So this is a proof of concept that we're also pushing forward. In the meantime, though, what we're actually doing, according to classic bioassay guided fractionation, is we're going through an iterative process of partitioning, fractionating, retacking, prep LC, mass spec, database dereplication, NMR, and even down to X-ray crystallography. One of the hurdles that we ran into early on in the lab is our ability to fractionate large amounts of isolated compounds. I just want to highlight this. This is a paper we just submitted. It is for a robotic system that we developed out of Legos and wood. I'm not kidding. Under 500 bucks, we're able to build this system. It usually would cost between 10 and 20 grand. And so very exciting. And this has enabled us to basically program our separations by HPLC and run 10, 12, 15 iterative runs over and over again. And what's happening is that we're getting so much of these isolated compounds in these flasks. When we dry them down, we're actually able to get individual crystals of some of these compounds. Here's an example. So this is just about ready to submit. We've discovered these bioactive triterpenoids. These are known compounds, so their activities have not been reported before. And what's interesting is that 2 of these we were able to get crystal structures for by X-ray. And when you put these individual compounds back in the mouse, we know that from the bioactivity side, their bioactivity is not that different in vitro, but in vivo, we see clear advantage to compound number 2. So that was another interesting finding. Here we have 3 molecules that behave quite similarly in vitro, but in vivo, there's one that seems to perform even better. So that brings up the question of, could there be some other host effects that are happening that we're not aware of yet? And that's a question we definitely want to chase down. So that's the staph toxin story. I'm going to move over to one of our other projects on how to overcome resistance through antibiotic natural product combinations. So we have some challenges where bacteria can resist antibiotics through decreased penetration, enzymatic degradation, blocking target sites and so on, or efflux pumps. And so what we're focusing on in this particular project is looking at enzymatic degradation. And our method is one that we modeled off of a method that Eric Brown's lab was using actually with an FDA, the FDA's approved drug library. And instead of using that library, we're using our own and are combining, basically that's what the QMPL is, the Clean Natural Products Library. We're combining our extracts, we're testing them alone and with a quarter of the MIC of the antibiotic and bug combo we're going after, and then we're following up with 2D checkerboard assays. And so the idea here is that we're looking for compounds or natural products that can enhance or restore the activity of antibiotics in a synergistic way to overcome these resistance hurdles. So I'll give you two examples. I've got another staph example for you. So the first here is with methicillin-resistant Staph aureus, and in this case, it's really exciting. This is with an extract still. We've done more work. I can't reveal what the compound is yet because we have to file patents and all that jazz on it. But what's exciting is that even with the crude extract, we were able to drop the MIC by 1,000-fold with peroxycelin. And we've pursued this through bioassay fractionation and are working on wrapping up this particular case. And this is from a plant that was used by the Seminole and the Creeping Muskogee. It's a southeastern plant that you can find here that has this interesting activity. We've also tried to apply the same approach to our work on Acinetobacter baumannii, carbapenem-resistant strains, and have found some limited activity. This is just proving to be a really tough nut to crack because Acinetobacter has so many different neolactamases, and it's like even if you're hitting one, you're going to have 5 others that step in and take over. So I'm not sure that this is really going to be the right approach for that bug, but we did have much more luck with staph. Another example of our more classic antibiotic and antifungal discovery work, again, I can't reveal what this plant or compound is yet, but we do have a structure on this compound, is that we took this from a plant we're looking again for our A. baumannii study just for growth inhibition, and you can see in this parent-to-child tree, again, using this iterative bioassay guided approach, and we can reproduce this approach through iterative runs. So we're able to closely follow the recipe and get more and more of the compound. And just by tracking it, you can see that we have these, the isolation, and these 3 fractions actually have some of the same compound in it. Interestingly, while this is active against A. baumannii, and here you can see the activity curve, we have an IC50 around, I'm sorry, we have, this is the isolated compound in the red, this is the extract in blue, and so the compound alone, we have an IC50 of just under 10 micrograms per mil. That's pretty good for Acinetobacter, especially reminding you this is a hyper, carbapenem-resistant strain. That's pretty good. And then we've also expanded our work to looking at Candida albicans and other non-albicans Candida species like Parasolopsis and glabrata, and Candida auris. Anyone heard of Candida auris? Right, so this is an emerging fungal pathogen threat. Mortality rates are as high as 60% for patients infected by it. The serious emerging threat that is also multi-drug resistant is resistant to 2 of the 3 fungal classes of drugs. And so this had MIC of 0.125, really nice MIC. So, and we also tested these in counter screens against human skin cells, and a decent therapeutic index of takes around over 100 micrograms per mil to get toxicity compared to under 10 for the bioactivity. Okay, next story, biogums. So when you wake up in the morning and you rub your teeth with your tongue, that slimy, gritty kind of feeling. That's what this looks like under a microscope. So you have these polymicrobial communities that love to stick to surfaces. Just like that staph got into my bone and stuck inside of my bone tissue, you also have biofilms that can pop up in other places in the body. Biofilms are challenging to treat because they are intrinsically resistant. It's difficult to get antibiotics to penetrate through all of this polysaccharide matrix. It's also difficult to act on these because antibiotics work on rapidly dividing cells, and these slow down their metabolism. So they have a number of interesting hurdles. And so some of my early work, this is work I did when I was in Mark Smeltzer's lab at UAMS, was to look at and identify the molecules responsible for biofilm inhibition in the elm leaf blackberry. And so this is a plant I documented as being used in traditional medicine today by people in southern Italy to treat skin abscesses and infections. And they would report using the leaves of this plant mixed with some pig fat, put that onto the skin, and if you're wondering why the pig fat, that can also help with absorption across the skin barrier of some of those molecules. And so I was really curious about what could possibly be responsible. This is an example, again, of another plant that had no growth inhibitory activity against Staph aureus. But here in this confocal microscopy, you can see that at 50 micrograms per ml, you have very limited attachment to surfaces. And here you have, at lower doses, you have some more attachment and a nice thick towers that form. So in one particular study, we took segments of IV catheters, so these are the same IVs that you put into your arm in the hospital, and we coated them with a strain known as UAMS1. We had a control strain, UAMS 929, which is a mutant, the SAR-A mutant, which is important for Staphylococcal biofilm formation. And we grew these up with and without antibiotic, and with or without the biofilm inhibitor, and with antibiotic and biofilm inhibitor. The first finding that was disappointing was that if you already have a preformed biofilm and hit it with 220-DF2, this extract, there's no difference over control. If you look at the antibiotic, this is 10 times the normal dose of the MIC of daptomycin. You have some decreases in adherent cells, but when you combine dapto with this extract, then you start to see a real impact after several days of treatment. And so we repeated this experiment with a number of different functional classes of antibiotics, and we saw the same trend where you're seeing an improvement when you combine the extract with these antibiotics, but it's not immediate. It takes several days to get these to go off of the catheters. So one challenge that we came into right away was that as I tried to separate this and follow that beautiful fractionation pathway, I lost activity. Over and over again, I kept losing activity. And I was just banging my head against the wall. I didn't know what to do. So what we did is we used mass spectrometry to identify the putative matches for the molecules in that extract. And it turns out that it was very rich in these compounds known as ellagic acid glycosides. And so to produce enough ellagic acid glycoside to test these individually and in combination, I worked with a synthetic chemist at Emory. She's now at Penn, Dr. Emily Weinert, and she was able to build these molecules for us in her synthetic chem lab. And we were able to test it. And what we found was that there was one particular derivative that was more effective with the ellagic acid rhamnocide, but even that was not as effective as the overall composition. And this is an important point. One of the strengths and limitations of working with botanicals is that sometimes the best activity comes from synergistic mixtures. I think that's important to note because nature works through synergy. Nature does not say, aha, I'm going to make this one compound and this is what's going to work. When we pluck those one compounds out, for example, with artemisinin, everyone familiar with artemisinin? Anybody? One person? Two? Okay. Oh, this is like the Nobel Prize in Medicine was awarded for the discovery of artemisinin. You guys should know this. Just a few years ago, too. Right? It's from wormwood, and they plucked out this compound, and we don't see single therapy artemisinin anymore in malarial treatment because you have widespread resistance. But if you look at the whole plant extract. There have been some beautiful studies published in PNAS that have shown that you can restore activity because it's that mixture of compounds in the plant that makes it most effective. There are a couple of different avenues you can pursue, one of which is the FDA's botanical drug pathway, which allows for these compositions to be approved as a drug. There are two examples of this that have made it out on the market. One is crofelemer, which is an anti-HIV diarrhea medicine from the dragon's blood tree in South America, and the other is green tea extract. It's actually topically applied for anal and genital warts. But you never thought your tea could do that. But it's been FDA approved for that. What are we doing? I'm not making wart cream, but I may in the future. You never know. So right now, what we're doing is we're taking this extract from blackberry that we know works in combination with antimicrobials. We're combining it with lemongrass, and lemongrass essential oils have broad spectrum antimicrobial activity. And we're developing this as a medicated bandage to treat patients that have ulcers and non-healing wounds. If you've ever been into a nursing home and seen how the elderly can often develop these bed sores that are difficult to treat and difficult to heal, they also smell really bad. So the lemongrass is also selected in this case for the better smell of the wound bed environment. And this, we're seeking FDA approval for this following the medical device pathway. So in conclusion, is there a role for botanics to play for in solutions for AMR? Yes. Yes. It's a participatory exercise. Yes. Will all effective botanical and natural products for infection control act in the same way as classic antibiotics? No. Right. Good. And lastly, is it possible to work with, this is a long one, sorry. Is it possible to work in systems of traditional medicine to determine which therapies are effective and deployable to a broader population, while also ensuring that they benefit from that economic development? Yes, absolutely. So that is my story. I'm sticking to it. This is my awesome team. I'm just the performer. They are the ones that are doing all the amazing work. And we have an amazing network of collaborators, both in the U.S. and abroad. Most of the work I presented today was done in combination with Alex Horswell and Lynn Weinert, Dan Zarowski at Rare. Some of this was done with Julia Kubanek at Georgia Tech, John Baxta, and a lot of field studies were done with these guys, with Alessandro, Andrea, Alfonso, Avi. These are some of the lead players from our research team that contributed these studies and our funding agencies. I'm also going to just leave a little plug at the end for my podcast. If you're interested in plants, pharmacology, and food, you should tune in. And thank you all for your attention. Questions?
So I worked in green natural products for the past 3 years, and we had a really hard time getting a greater amount of active compounds. Because it's hard to get sponges from the ocean, and it's hard to
Not hard to get temperature.
Yeah. That was my question. Is it's much easier with plants to grow more and to obtain more extract.
I think one of the greatest myths, and this was kind of perpetrated by Hollywood. If you've ever seen Medicine Man with Sean Connery, right? Where the cure is this elusive orchid at the top of the forest. No. People use plants that are easily accessible to them, and in many cases, many of these medicinal plants are very weedy. Weeds are interesting to me because what is a weed other than a very successful plant? If you're a very successful plant, you're gonna have some interesting chemistry at play. Same thing for witchy plants and magic plants. Magical plants, a lot of plants that were assigned magical attributions, those turned out to be sources of some of our greatest cancer drugs. Because people in their own way, in their own culture, might attribute some interesting or toxic activity to spirits or whatever, but in reality, it's the chemistry.
Yeah. First off, I just wanted to thank you because I thought it was a really awesome talk. And you said kind of in the beginning that it was important that your collaborations with local communities are kind of like a two-way street, so they derive some benefit from it as well. I was wondering, do you see a place for your research and the work that you do with local communities in like bolstering conservation efforts for those communities? Because it could be argued that like if all of these, you know, like environments that are under threat, if they're big untapped sources of new antimicrobial drugs, you have a big argument for conserving natural spaces.
I mean, I think that's the hope. And I mean, I also, there's again also this mythology of the primitive native man living in the forest, and you know, it's like these are real people that have families that need to make money, that need to have income, that you know, they need to be able to make a living just like us. And so I like to think about how can we couch this kind of work also within an economic framework that can bolster communities. So, but in a sustainably harvesting kind of way. So it's definitely in my mind. I'm not an expert in development, but I hope that this will, you know, these kind of things, you really need people that are experts in development and economics to jump on board, you know, and make those things happen. So that's something like, that's definitely a dream of mine to make that happen.
So when it comes to animals in agriculture, I mean, animals have been known, if you observe their behavior, they can eat plants when certain health, yes, you get what I'm saying.
Yeah.
And they have certain health outcomes. Have you looked at the use of any of these compounds, or have you observed in Central South America any of these animals eating certain compounds that you might be looking at for both human and both animal and veterinary uses?
Yeah, that's a great question. So you just described the practice of zoopharmacognosy. Zoo for animal, pharmacognosy is knowing of drugs. And there are definitely primatologists and people that specialize in observing animals that can contribute to this field. I know nothing about how to observe what an animal is doing. I don't sit there for, I mean, these are people that dedicate their lives to really observing animal behavior. So I think that's something that they could definitely contribute to the field. And they already have. In many cases, there are some great examples of that. What also is noteworthy is that humans also engage in something known as ethnoveterinary medicine, which is they actually will treat their animals, their livestock, also with medicinal plants. And what's interesting is sometimes they'll use remedies that they may not prefer for themselves because they have a burning sensation or they aren't very comfortable to use, but they will put on their animals out of necessity. And remember, these animals, for example, in areas where I work in Albania, those livestock are essential to their survival. If their horse is unhealthy, if their mule can't work, if their oxen can't plow that field, their family could starve. I mean, so care of animals and kind of veterinary practices is definitely big there. But yeah, it's a good question. There's a lot we can learn from the animal world, for sure.
I was wondering if you've tested your library of natural extracts for cytotoxicity against host cells, like at the concentrations that you're seeing working against these bacteria. Like, I'm especially interested in the escape pathogen.
Yeah, absolutely. So we haven't done work on macrophages yet, but we have done, we always counter screen against keratinocytes. We're probably going to try and expand that to include also like liver cells and things like that. But right now, all of our studies have been in topical animal models. So we're observing, you know, we're watching for any kind of toxicity in the animals. But before that, we're also testing in human keratinocytes by LDH assay. And so I mentioned before therapeutic indices. We're looking for 1 to, at a minimum of 1 to 10, but hopefully more than 1 to 100 in terms of dose differential and IC50s. But again, this is also a space where collaborating with medicinal chemists can really come into play, and they can help tweak that blueprint, right? Because maybe what we're isolating is not the perfect drug, but it's a starting point. So there are ways to modify and help improve those different dynamics.
You see that there's like fewer amounts of cytotoxicity in the compounds, like against the keratinocytes, because they're natural extracts, or?
So I'm going to be careful in how I phrase this. First of all, not everything that's natural is safe. A lot of things nature that would kill you. Okay. I think that because our library is targeted to plants that are being used by humans already as topical anti-infectious agents, that we see less toxicity typically in those extracts because people have selected them. They're not causing like severe reactions. We have had cases where we've actually had horrible toxicity. I told the students earlier today, I think at lunch, you know, I have a collaborator who's a mycologist in Europe, and he sends me all kinds of cool fungi to test. And he says this one mushroom, and it had a beautiful purple extract. It's gorgeous. I was so impressed by its color. And we were running the acne and Acutibacterium acnes campaign, and man, this one just knocked over. It was awesome. Killed it. Got rid of biofilm that was already established. I was so excited. We found the golden ticket acne cure. Then we put it on the hackets and also killed all those. So we had a full face peel on our hands. So yes, there are lots of examples of things that are natural but are also deadly. And remember, many of our medicines, though, are poisons, just at a different dose.
The different types of honey, different antimicrobial activities, partly because they come from different types of pollen derived plants.
Yeah.
Did you ever look at the antimicrobial activity of honey, try to look at which plant species it came from?
Yeah, so it's a great question. So yeah, there are medical devices that have honey embedded in them for wound care. Honey has become a really big thing in wound care. So interestingly, because of some press that we got in Nat Geo on our pepper tree story, I was contacted by a beekeeper in South Florida that collects a special pepper tree honey every year. And they sent us some of the samples to test. And unfortunately, the compounds and the bioactivity just wasn't there in that particular honey from what we observed with the, But we haven't really looked at the other kinds of medicated honeys in depth. Chemistry is a little bit nasty. They like to mess with all the sugars out.
Sorry, one last. So I know a few examples where people thought that a plant was medicinal, and then actually through further research, they find that a microbe that lives in the plant is the one producing the compounds like Taxol and Nutri is actually made by fungal endophyte. Does that present any challenges in your work, and do you do any work in your lab to kind of like pick apart where the molecules are actually coming from?
That's a great question, and we're very aware of this, like the onion that peels, pulling back layer after layer. So we don't currently culture fungal endophytes. There's an amazing lab group that does at UNCG. There's Nick Overley's and Naja Czech's group. They do this exceptionally well, and they have in-house mycologist that basically identifies all these, and they're pulling these out of different medicinal plants. It's really exciting work. But if I were to add that on to all that I already do, I think we'd stretch ourselves a little bit too thin. But it's something I'm very aware of, and as things advance, especially as we look for ways to scale up production of some of these bioactive molecules, if we can't do it via synthetic routes, that's definitely a route to also explore. And in the end, I think when it comes to traditional medicine, whether it's coming from an endophyte or the plant itself, the point is they have the knowledge to know where to find this remedy. Yeah, that is another interesting element. One last question? Yeah.
I'm curious, so when you're extracting from these plants, you're growing them in the lab, or you're taking them from the lab?
We wild harvest everything.
Oh, wow.
I have a brown thumb. It's embarrassing. Like I cannot grow plants. I've killed so many plants in my office. Like I had to carry a palm tree out there.
It's interesting. When I worked with marine natural products from marine bacterium, it's like depending on the growth conditions, I got different compounds.
Yeah.
And so it seems like stress would induce more production of
So
compounds.
that's a good question. So we do recollect at the same field sites, or every area where we collect, we have GPS coordinates, we have information on dates when we go back. And so when we have things of interest, we might go back and recollect the same site for a different period of the year. We just did that in South Georgia 2 weeks ago. So we did a June collection, and we just did another collection of some of the same species. So we also looked at like predator enclosure exposure studies, working with some of the ecologists there to see if there are differences with like over foraging on the populations. But yeah, those are definitely aspects that influence the chemistry. The nice thing though is that with the use of marker compounds and these standardized techniques, and you know what you're looking for, you can create batch to batch, very rigorously controlled batches of refined chemical product, regardless of whether or not your plant produced more or less of it during that period of the year. But that is an extra area of complexity we have to think about in our process. Yeah. Well, thank you guys so much. I appreciate it. If you have any other questions, feel free to come up and chat. Thanks.